So folks, the last thing that we're going to discuss briefly is that some enzymes require molecules called cofactors in order to be functional. Now, what are cofactors and why is this, right? So, up until now, what you might be assuming is that the amino acid R groups of enzymes are performing the chemistry, and you would be right. R groups can do a bunch of types of chemistry. They can do acid and base chemistry. They can perform ionic interactions that are important for the chemistry to happen. They can perform transient covalent bonds, so they can form bonds, they can break bonds. And in this case, it's the protein itself, it's the R group that's doing the chemistry. However, we're really limited to those types of chemistry. And in our bodies, we need to do so much more chemistry, folks, right? In the previous slide that I showed with all the different types of chemistry, for example, R groups really aren't going to be doing oxidation reduction reactions. So, in comes cofactors. So cofactors are molecules that perform the types of chemistry that protein cannot. They can be non-covalently associated with the protein, or they can be covalently associated with the protein. But they are required for the activity of the enzyme, and in this case, it's the cofactor itself that is the catalyst and performs the chemistry. A little bit of new vocabulary here, but it's not going to be a huge focus, but so you can understand this language. When an enzyme uses a cofactor but is not yet bound to that cofactor, we call it the apoenzyme, and it is inactive. It cannot function. Once it binds its cofactor, either non-covalently or covalently, we call it the holoenzyme. It is active, it is complete and whole. And now let's look at a few different examples of cofactors, because they can be pretty different from each other. A great example of a cofactor is a metal ion. Think copper, think iron, think zinc. There's a picture here on the right, folks, of a protein that uses an iron-sulfur cluster to help it perform its chemistry. Iron-sulfur clusters are very common, and we will see an example of many proteins that contain iron-sulfur clusters, when we discuss the electron transport chain. Now, because some enzymes require metal ions, this is why, unfortunately, heavy metal poisoning is a thing. When the ions in your enzymes are replaced by other heavy metals that aren't supposed to be there, those enzymes can lose function, resulting in some of the effects of heavy metal poisoning. But cofactors can be more than metal. They could just be organic molecules, small organic molecules or larger organic molecules. And there's some more vocabulary associated with these types of cofactors that we won't worry about, but so it's here on the page for you. If that cofactor is really tightly bound to that enzyme, sometimes we call it a coenzyme because it's really a part of that enzyme. If this cofactor comes and goes, right, it comes in for the reaction and then leaves, a lot of times we'll just call it a co-substrate because it's almost behaving as a substrate. Two examples of this are NADH and NADPH that we'll talk about more when we get to metabolism. So here on the bottom, I'm showing you the reaction performed converting ethanol into acetaldehyde by alcohol dehydrogenase. And you'll see that since the reaction being performed is an oxidation, we need a cofactor, and we'll use this cofactor called NADH that we'll learn about later in this class. And what you didn't already know, because I didn't mention this, is we saw an example of a cofactor already in this course. However, we didn't call it a cofactor because we saw it in the protein myoglobin and hemoglobin. So since those proteins do not have enzymatic activity, it's not necessarily correct to call it a cofactor, but that heme molecule that is used in hemoglobin and myoglobin is a similar molecule used in many other proteins that do perform oxidation reduction. And so these types of prosthetic groups or heme groups are also an example of a cofactor, and as we saw with hemoglobin and myoglobin, are often covalently linked to the enzyme. That wraps up our quick introduction to enzymes, folks. We learned that they are biological catalysts. We talked about the different types of enzymes that exist. We got a brief introduction into binding of enzyme to substrate, and then we talked about cofactors there at the very end. With that, I'll leave you with this cat meme. See you in class.